Thank you very much for the introduction. Yeah, indeed, my name is Merel van der Thiel. I'm working in Maastricht, also together with the Alzheimer's Center in Maastricht. And our work is mainly on the waste clearance system of the brain. So Adam already touched upon it a little bit, but I will start off with some background on this waste clearance system. So cerebral waste clearance reduces with healthy aging and in various neurodegenerative diseases, for instance, in Alzheimer's disease, where one of the theories is that toxic proteins such as amyloid beta are not properly cleared from the brain, which causes toxic aggregates and subsequently causes dementia. And the clearance system is thought to work through a network of perivascular spaces, where fluid runs parallel to the arteries through the perivascular spaces, then flows through the interstitial fluid, flushing away soluble waste products, and is then washed out again on either the side of the venous or the arterial perivascular spaces. Therefore, perivascular spaces play an important role in the waste clearance. Literature has suggested various drivers of this clearance system, or more specifically, of the movement of interstitial fluid within the perivascular spaces. And one potential driver is the cardiovascular pulsatility, where pulsation of the vessels drive the movement of the interstitial fluid within the surrounding perivascular spaces. But to investigate the effects of pulsatility on interstitial fluid motion within the perivascular spaces, a measurement of pulsatility is needed for the very small cerebral arteries, together with a measurement of interstitial fluid movement. But the small scale of these phenomena make it challenging to investigate this in humans. So we used ultra-high field MRI at 7 Tesla, which enables to achieve a very high spatial resolution, and thereby allows the visualization and the measurement of the blood flow velocity waveform in very small cerebral vessels, using phase contrast MRI. This could be done, for instance, in the lenticular arteries, or the LSAs, which branch from the middle cerebral artery and are in close contact with the parenchyma of the basal ganglia. The blood flow velocity waveform can then be calculated and can be used to calculate the pulsatility index, or the PI, which is considered a measure of arterial stiffness or compliance of these lenticular arteries. And to be able to get a measurement of interstitial fluid on this very small scale, we use a diffusion technique called Intravoxel incoherent motion. So Intravoxel incoherent motion, or IVIM, is a diffusion technique which provides the opportunity to estimate diffusion components on a microscopic level. So traditionally, the IVIM model is used to estimate a parenchymal and a microvascular component. But the spectral analysis method using non-negative least squares does not lay any constraint on the number of diffusion components that can be estimated from the IVIM signal. And the uses of this method thereby led to the identification of a third diffusion component, which is thought to be representative of interstitial fluid. So this component is thought to represent interstitial fluid properties either between the parenchymal cells or within the perivascular spaces. And when looking at the spectrum, this component lies between the two traditional components. Therefore, it's called the intermediate component. And the diffusivity of this intermediate component, dInt, has previously been found to be associated to both vascular and neurodegenerative markers in a memory clinic sample. And additionally, previous work has found higher dInt values inside the perivascular spaces as compared to the surrounding tissue, which thereby highlights the sensitivity of this measure to properties of interstitial fluid. And a region that is specifically known for the presence of the perivascular spaces are the basal ganglia. And these are gray matter structures that are located in the deep gray matter, and as mentioned, are supplied by the lenticular arteries. Therefore, this study took the two measures together and aimed to gain more knowledge on how variations in the lenticular artery pulsatility, as measured with phase contrast MRI, relate to dInt derived from the basal ganglia, as measured with IVIM. So we foresee to find that stiffer arteries, which is reflected by a higher velocity pulsatility, are associated with impairment of the surrounding tissue, which is represented by alterations in interstitial fluid diffusivity. We investigated this in 25 healthy elderly subjects who were scanned on a 70 Siemens scanner at a Scanexus scan facility in Maastricht in the Netherlands. And we acquired an anatomical T1 and a time of flight angiogram for the planning for the phase contrast LSA scan. And for this phase contrast scan, we measured a minimum of 16 cardiac phases and a VENC of 30 centimeters per second. And using the 7 Tesla, we were able to reach an in-plane resolution of 0.31 millimeters. And the diffusion data for IVIM was acquired in 3 directions, and we applied CSF suppression. And as can be seen, the IVIM sequence mainly focuses on low B values. On the time of flight images, a cardiac-gated phase contrast MRI was planned perpendicular to the lenticular arteries, as you can see here in blue. And the largest LSA was processed to assess the blood flow velocity measures. So after correction for background noise and aliasing, the vessel area was quantified from these magnitude images. And from this, the blood flow velocity wave was calculated, from which the pulsatility index, or PI, was calculated using Gosling's equation, which means subtracting the minimum velocity from the maximum velocity and dividing this by the mean velocity over the cardiac cycle. So this resulted in a pulsatility index measurement of the largest LSA within the basal ganglia for each subject. So the IVIM images were processed in the following manner. They were corrected for motion, eddy currents, and EPI distortions. And the 3 directions were then averaged into 1 trace image. And then we performed regularized spectral analysis in the native IVIM space using non-negative least squares to obtain a d-int value for each voxel. And the intermediate component was identified by the borders that are shown on the slide. So we used FreeSurfer to segment the basal ganglia from the anatomical T1 weighted images. And the median values of the interstitial fluid diffusivity were extracted from the basal ganglia ipsilateral, so on the same side as the post-process LSA, which allows us to really study the local effects of the LSA pulsatility on the interstitial fluid in the surrounding tissue. And we use partial Spearman-Rowe correlations to investigate the relation between the PI of the LSA and the interstitial fluid diffusivity while adjusting for age and sex. So we found that a higher pulsatility in the LSA significantly relates to a higher interstitial fluid diffusivity in the nearby basal ganglia tissue, which was independent of age and sex. So this relationship can also be seen on the example on the right, where a high interstitial fluid diffusivity in the basal ganglia, as is shown by the lighter yellow, relates to a higher pulsatility index, as the velocity waveform of this person shows a large difference between the minimum and the maximum velocity over the cardiac cycle. Likewise, you can see an example of the interstitial fluid diffusivity map of a person with a relatively low interstitial fluid on the lower row. And this person also has a lower difference between the minimum and the maximum velocity, and thus would have a lower pulsatility index. So the current study found that a higher pulsatility within the LSA is related to a higher interstitial fluid diffusivity in the nearby tissue of the basal ganglia in an elderly population. So a higher LSA velocity pulsatility may reflect reduced dampening of the upstream vessel wall caused by increased arterial stiffness. So when perforating arteries momentarily dilate, as you can see here on the left, the perivascular spaces that surround this momentarily become narrower. But when the vessel wall becomes stiffer, as you can see on the right, this short-term dilation and also the narrowing of the perivascular spaces is reduced, which results on average for more time and space of the water molecules inside the perivascular spaces to diffuse, resulting in increased interstitial fluid diffusivity. So to conclude, increased arterial stiffness in the very small cerebral arteries seem to be associated with increased interstitial fluid movement, potentially in the perivascular spaces in a healthy aging sample. Furthermore, this study forwarded the IVIM-derived interstitial fluid diffusivity as a potential non-invasive proxy of alterations in perivascular space fluid movement. And using this non-invasive technique, more knowledge could be gained on the effects of vascular alterations on waste clearance, which has applications for many neurodegenerative diseases. So thank you all very much for listening to this presentation, and feel free to reach out to me if you have any further questions.